Have you ever thought about how you can see, why you can see, the reason behind your vision? Throughout time, species and animals have adapted to their ecosystems and environments through evolution. They see the world through their own perspective and different ways, as they all view the world differently. So, my project is gains and losses to trade-offs in eye development. As Ryder said, I worked at LSU over this past summer and did research in a lab. So the first fish I want to talk about is the four-eyed fish, or its scientific name, the A. anablet, but I'm just going to be calling it the four-eyed fish. So this fish, over time, had 2 eyes, and its pupils split into 4 pupils. So it has 4 pupils for 2 eyes, and it can see above and below the water simultaneously. The second fish I want to talk about is the blind Mexican cavefish. This fish lives in dark underwater caves, and over time through evolution, its physical eye deteriorated into just a retinal area. So it can't see, but it still has some reminiscence of a physical eye. So I did my work in Dr. Schneider's lab this past summer. She specializes in the research of these four-eyed fish, and I got real college lab experience, and it was a wonderful experience I got to do this past summer. I got to work with undergraduate students and graduate students, and it allowed me to just really get a sneak peek into what college level research is. So, come on, there we go. So, for my project, it's kind of a 2-part thing, really. So the first thing is that I wanted to look into how the Mexican cavefish has evolved over time. So I wanted to take a look into its retinal area. So my first hypothesis is that the Mexican cavefish possesses little to no form of neuronal activity inside its retinal center. So over time, it has completely lost its form of retinal, any neuronal activity happening inside its retinal area. And the second thing I wanted to do was compare the 2 fish together, so the heightened sense of vision four-eyed fish to the blind Mexican cavefish. So the first thing I did, this was special preparation, so if we would euthanize the fish, so we'd kill them, and then we would dissect them for their brains and their eyes, and then for the Mexican cavefish, we'd have to cut the head off so we could be able to slice up the retinal region. And then for storage, we would then freeze them with a substance known as tissue tech, and then we would freeze that with liquid nitrogen, and then we would store it in a negative 80 degrees Celsius freezer. And then for cutting it, we used a machine called a cryostat, which slices these frozen block samples into 5 to 10 micrometer samples. And they're very tedious to work with, especially on an 80-degree day inside the lab on a hot Louisiana summer day when the cryostat's negative 20 degrees Celsius. It can be very annoying to work with. But once we do that, we put them onto these slides. And for each sample, we can get up to about 100 slides with about 3 to 5 samples per slide. So we have a vast amount of material to work with. Viewer discretion, I'm about to show some pictures of some fish brains. If you don't want to see that, look away. So the first one this is a dissected four-eyed fish brain. So right here is the brain stem. This second portion here is the optic tectum region of the brain. This is where the retinal neurons will be relaying information to, and then you have the frontal lobe areas. And then the next one is the cavefish brain. As I said, like the brain stem is here, the optic tectum and the frontal lobes here, as they share a common ancestor of the zebrafish, which is why a lot of their features stay the same, but they're also vastly different in their many ways. So what we did was, with these slides, we used various slide staining methods to be able to view these slide samples under a microscope. And we used these in ways so we could easily extract information. So the first method we used was H&E staining, which is used as a checkpoint when we're using the cryostat and slicing samples. So basically we use that to kind of see where we are in the sample, to kind of get an idea of like where we are. The next 2 are Nissl staining and fast blue staining. Nissl is used to look at the structure and integrity of the sample, and fast blue staining is used to identify nuclei and see if those are still intact on if those are still intact on the sample. Then the most important one is immunofluorescent staining. This staining process uses proteins and antibodies to help identify photoreceptors, neuronal cones and rods, and retinal nuclei on these cells and to view under a microscope. And in this picture, this is what I'm doing. I'm pipetting the antibody solution onto the slides, which are inside this box right here, and they would sit there for about 2 days. So here's some examples of what the slide staining looks like. Right here is, in this box, is the H&E staining. This is of the cavefish brain. So in this portion of the brain, this is part of the optic tectum region. So this is a very important part of the process. So that would tell us that we're in a very important part, and we need to be very careful when we are cutting the thumb sample. And the next portion up here, as I said, they're normally used hand in hand in the nissle, which is on the bottom here in the purple, and the fast blue in the blue up top. So this is a zoomed in portion of the brain of the cavefish. So this would show that it's in good structure, and the fast blue shows that we have lots of nuclei there. So it's a very good sample to use. So that tells us that the surrounding samples in this whole data set that we have are ready to use for immunofluorescence. So here are my results. So these are the immunofluorescent images. You probably can't see them too well. They're pretty dark, but I'm going to explain them best for you. So here in the blue, in this box, is called DOPI. This, we are able to see photoreceptor cells and nuclei. So this will tell us like what the amount of cells that are still in this area of the region. And in the green, we call it ZPR1. This will actually tell us what kind of activity is happening inside of the region with the neuronal, and it identifies those cones and rods, as I said earlier. So as we can see with the DOPI staining in this box, that there is a decent amount of cells here, but in the green box, there's not any activity happening. There's about 2 to 3 specks here, which is basically nothing. There's nothing happening here. And then we're going to move on to the brain region. So this is a zoomed in image of the optic tectum region of the cavefish's brain. And this, again, is the DOPI, which shows us the nuclei that are in relation to the retinal cells, that from the neurons that connect from the retinal area to this portion of the brain. So we have a lot of nuclei in relation to the retinal area in the DOPI, but in our ZPR1, which would reveal the retinal activity, there's not a lot. There's basically, like earlier, there's basically a speck or two inside of this area, which again shows that there is basically no activity happening. And then this is an overlay of the two images, which looks really good, and it allows us to kind of get a better image of the two, so we can really see like what is happening with the activity compared to the nuclei. Now this brings me to the second portion of my project, in which it is comparing the two extreme ends of these fish who have derived themselves from evolution. So here, again, we're using ZPR1 and DOPI as that is in this overlay. In this box to the left is the four-eyed fish. This is the eye here, and this in this box is the retinal region of the cavefish. So in the four-eyed fish, we have lots of photoreceptor cells along with the neuronal cones and rods seen here in this green portion of the area along the basic structure of the eye. And this shows that this eye from the four-eyed fish has a heightened sense of activity and there's just a lot of stuff going on inside this eye, which shows its enhanced vision. And then for the four-eyed fish, obviously, there's nothing going on. I mean, not the four-eyed fish, the cavefish. There's obviously nothing going on as it is blind. So as we can see, there's not a lot of photoreceptor cells, as there's not a lot of blue, and there's barely any green. These green splotches up here are from dirt from other sediment that could have gotten onto the slides. But in the important area, there's no green here at all. So what this means is that the cavefish doesn't have any activity happening in its retinal region, but with future research, we can maybe see where maybe these neurons have rerouted somewhere else. How are they enhancing some other function inside of this cavefish? And it's just really cool to just look at all this and see how these fish have evolved from their common ancestor of the zebrafish. And I just want to say this: we're all born from differences. I have been able to compare species that have come from a single point on the evolutionary chain, as these species have adapted to have the best chance for survival. And I think the most important and most interesting part of this project is the fact that these species have left the door open for change. As like I said, the cavefish could still have neurons that could have went and enhanced something else, and maybe if its environment had maybe changed. And I just want to say this, the best way to summarize my project here is from this quote from the movie Jurassic Park: "Life always finds a way." Thank you.